Right now, about 1 trillion blood platelets circulate through your body. They are incredibly small. An individual platelet has a volume of just 10 femtoliters, roughly 100 millionths of a cubic millimeter. These cells act as the primary guardians of your vascular system. When a vessel is damaged, platelets adhere to the exposed walls and aggregate to form a protective clot. That clotting mechanism is highly sensitive to its environment. For astronauts in orbit, microgravity alters blood behavior, which can trigger obstructive thrombosis or severe hemorrhages. When a platelet activates, it morphs. It stops being a smooth, oval-shaped disc and extrudes chaotic, irregular spikes outward from its surface. That biological shapeshifting is what keeps us alive, but its unpredictable geometry creates an enormous roadblock for researchers trying to model it. Observing platelet behavior under the intense high-shear forces of active blood vessels is nearly impossible under a physical microscope. Researchers have to rely on computational fluid dynamics, or CFD, to digitally simulate how blood moves. To run those simulations, CFD algorithms require one strict input: predictable, precise mathematical geometries. The software needs clear coordinates to calculate how fluid moves around an object. A smooth, unactivated platelet is easy to map. An activated platelet, covered in non-spherical, wildly irregular spikes, is not. Because the software cannot interpret the shape, simulation efforts in hematology stall. To move forward, mathematicians had to find a way to write an exact equation for a biological anomaly. The first attempt to solve this involved flattening the platelet into 2 dimensions. Researchers placed an image of a platelet on an XY axis and used polynomial interpolation to draw a mathematical curve tracing the outer edges. The math failed. Standard polynomial equations cannot plot perfect vertical lines, and trying to curve fit every irregular bump of the cell boundary created a bloated, inefficient set of calculations. The breakthrough in 2 dimensions came from a classic framework known as the generalized polar rose. The equation establishes an invisible central circle and generates outward radial projections that mimic the biological spikes of an activated platelet. By varying the length and width parameters of those outer petals, researchers can instantly generate infinite, mathematically defined cross-sections. Plotting a flat cross-section proved that the biological chaos could be constrained by math. But because actual human blood flows in full 3 dimensions, this 2-dimensional framework had to be pulled up into the physical world. The transition to 3D begins with a core Cartesian equation mapping a simple ellipsoid on an x, y, and z axis. To build the spikes, a logarithmic function is revolved around the z-axis, creating a smooth surface extending outward. In nature, activated projections erupt randomly across the cell. To replicate this, researchers apply rotational matrices, multiplying coordinates by pitch and yaw angles to twist the spikes into any direction. With a defined ellipsoid core, logarithmic spikes, and rotational matrices, researchers arrive at a functional 3D digital twin of a biological anomaly. To pull physical data from this new geometry, the digital twin is analyzed across 6 distinct orthogonal renderings, showing exactly how the same object looks across the x, y, and z planes. In this visualization, green numerical markers denote precise coordinate mapping. Researchers use these views to extract the exact spatial endpoints and physical widths of every single projection. Extracting the projected area and perimeter from these views allows researchers to calculate the platelet's drag coefficient, the exact measure of fluid resistance it creates. This measurement takes a static digital shape and turns it into a physics-ready object that will react accurately to the simulated flow of blood. Microscopic laboratory experiments on blood platelets are arduous and prone to physical error. Computational modeling replaces that friction with mathematical accuracy. With a reliable way to predict how activated platelets move and cluster, engineers can design customized medical stents and targeted antiplatelet drugs directly inside simulation software. By forcing biological chaos to obey strict geometric laws, scientists can bypass the physical limits of the microscope entirely, synthesizing life-saving treatments based on mathematical truth.